So I'm really happy to be here today to get a chance to talk to you guys about using biotech to feed the future. So we'll just jump into it. So my name is Amanda, and I'm a plant biologist. So that means I have spent all of my adult life, the better part of it certainly, fascinated with how plants work and how we can make them work better, which has really been the driving focus and questions that I've been asking. And so I'm coming to you today from the University of Essex, where we have the Essex Plant Innovation Center. So I have certainly found my people. You can tell my accent is not from here, but I traveled across an ocean and found people who love plants just as much as I do. And so the driving force for this is that I think it's not a huge surprise to anybody that's listening that the world has seen some huge changes. And so in the course of my lifetime, of course, we've seen changes in the global population. So while I have been walking the earth, we have had an increase of 60% more people, which has driven urbanization. Cities have expanded, right? Our footprint on the earth has expanded. And at the same time, that's meant that we've had to produce about 2% more crops per year. So our farmers have been actively engaged in this, growing enough food to sustain this increased population. But we also know that that's not necessarily enough at the moment. So we can look at this graph. I told you I'm a scientist. Scientists love graphs. And if we look at this graph, we can see yield versus years, so production of two of our staple crops, rice and wheat. And if we look at this, quite a few things in my hand. What I really want you guys to notice is that this solid line is showing us how much food we're producing from these two major crops. And we can track this back to about the last green revolution in the 70s, and we can see that we're increasing crop production, which is exactly what we need to do. The kicker to this graph is in the dashed line, because the dashed line is what the UN FAO tells us we have to do, or we have to produce, to meet mid-century population. And so that's a little challenge, or terrifying actually to me, because mid-century population is not that far away. When I started thinking about this, 2050 seemed a really far target, but 2050 is coming up pretty quickly. And so the real question that's driving a lot of research for plant scientists and a lot of the biotech research that plant scientists are involved in is how are we going to close this yield gap and how are we going to produce enough food? Of course, it's not without its challenges. So at the same time as we're producing enough food, we know that global temperatures have increased since pre-industrial levels about 1.2 degrees Celsius. I find this figure, of course, this is one of the show your stripes, really effective, because if, like me, you're a millennial, you haven't known that many cool years on the planet. And when I teach students now, they haven't known any. So they've only been living in the red. And so at some point, we have to wonder, how are we going to do this? How are we going to meet this yield gap while also coping with a climate that's becoming more and more challenging, right? Anybody that's lived through the summer of 2022 in this country knows heat waves are becoming far more frequent. With that is associated other climate stresses. And so we know that increasing crop production is vital, but we also know that as plant scientists, we're not necessarily going to get there alone. And so I really like this little figure because it shows us who we need to bring along with us, right? Scientists, certainly, we've been accused of many things. Definitely, many scientists will tell you someone at one point or another in their life has called them a bit of a know-it-all. But we know that we're not going to get here alone. And so we really need to harness support from crop breeders, farmers themselves, but also things like influencers, policymakers, politicians. And so it's not something that we're going to solve in our lab, in our field, running yield trials alone. And so that is why at Essex, we've established the Essex Plant Innovation Center. And so it's bringing together biology, but also technology and business. And so taking us out of our biological sciences home. I'm quite comfortable in the lab. I'm quite comfortable in the greenhouse. I'm much less comfortable talking to someone who builds robots because I don't know that much about robots. But we know that this type of automation is key to increase crop production. And so it's working to help us deliver solutions from the lab bench, from our sort of scientific home to business partners and policymakers. And so how can we do this? How can we actually help solve some of our yield troubles, how can we harness biology and technology to feed the future? And so I'm going to work you through two of the main ways that we're doing this at Essex. And the first is, of course, to make better plants. So biotech, I think many of you might have known, one of the things you were coming to get when you sat down here. But of course, making better plants is part of this. And one of the ways that we're doing this, and many of my colleagues across the world are doing this, is to look at photosynthesis. And so I love photosynthesis. I'm fascinated by the process. And many of you probably learned this many years ago and are thinking, God, this girl loves photosynthesis. How boring. But it's fascinating. It's actually 170 steps in photosynthesis, which really expands that equation that we all learned, you know, CO2, light, water makes glucose, makes water. No, it's 170 steps, and it makes life on Earth possible. And so just as a refresher, when I'm talking about photosynthesis, I'm talking about the process that plants use the sun, carbon dioxide, and water to fix this CO2 into sugars in the leaf. And then it's these sugars that are actually used by the plant for growth, but also to produce seeds often, the parts of the plant that we eat, and of course, our major foodstuffs. And so everything that we eat at some point has depended on photosynthesis. So if you had something to eat today, we can thank a plant, we can thank the photosynthetic machinery that brought the food to us. And so one of the key questions, if we're manipulating photosynthesis, right, if we're going to try to make photosynthesis better, is which one of those 170-ish steps do we start to manipulate? How do we know that photosynthesis isn't already doing a good enough job? It's doing the impossible. It's making sugars from air and water. So actually, the first way that we do this is that we start to measure it. So normally I would have our photosynthesis robot here, but it was a bit much to carry it in for the weekend. And so what we do is we clip a leaf into this chamber. And then with this chamber, we can set the parameters that determine photosynthesis. So we're thinking light, right? The plant uses light. Carbon dioxide, which we know is going up in the atmosphere, so we can set this to be realistic for now, but also the future. Temperature, humidity, and oxygen. And when we do that, we can figure out which of the factors are limiting photosynthesis at the moment. So is it getting too much light? Is it not getting enough light? Is photosynthesis going to be detrimentally responding to heat? Will CO2 help it? At what point does CO2 in the atmosphere stop helping photosynthesis? So these are all key questions that we've been asking. And once you get the answer to some of those, which has been an ongoing process for many years, what we can start to do then is to manipulate photosynthesis in the lab. So we just got some shots done in the lab, and I was quite proud of this one. Felt like I look very science-y. But really what I want to talk to you about is what it means when we use genetic modification to modify the process for the plant. Because I don't think this is something that scientists like me are particularly doing a good job of explaining. And so I'm going to walk you through the process of modifying photosynthesis from our last effort at this. And so to do this, we found a pathway that is part of photosynthesis that becomes more and more important as temperatures increase. And we thought, can we borrow some genes, can we use some genes from another organism to make a more efficient pathway? And so when we did that, we found the source of the genes we were interested in. So we found one from pumpkin and one from a green algae, Chlamydomonas. We isolated those genes using the pretty traditional molecular biology methods. We put those genes into a shuttle, so into another bacteria that's growing on the bench. You can manipulate everything with that. And then you introduce those genes back into a plant genome. And so in a lot of this work, we've used tobacco as a really quick model species. Because we want to improve tobacco? No. No, not really interested in improving tobacco. Not really a main goal of my research program or the research that the funders are interested in. But it is a fast-growing plant, and you can see it's got these really beautiful big leaves. And when you have to study photosynthesis, those big leaves are really important because you can put it in your chamber and you can measure it really well. And when we do that, we've found now we've manipulated 3 of those 170 steps in photosynthesis. So we've used 3 different strategies to do this. And the engineered plants on average grow 20 to 30% larger. Still in the model crop, of course, but you can see that here. So this is, we call our unmodified control. Scientists call it the wild type. And you can see, even in a public lecture, I haven't lost track of my scientists. And then these are alternative plants. So these plants have been engineered with those two genes from the green algae and the pumpkin. And when we grow them in the greenhouse, they grow larger. But it's important to note that this also happens when we grow them outside under various conditions. And so we see increased temperature resilience when we grow the plants. Because tobacco is a crop, we can take it from the greenhouse to the field. So we see increased temperature resilience when we put them under heaters. And so what you're looking at behind me is just a really beautiful photo of the heated and unheated plant canopies. And so it's just using a thermal camera. We've heated those plants up for their entire growing season in an agricultural field, 5 degrees Celsius. When we do that, we see that the plants are able to maintain more of their yield when they've been modified versus the controls. What we've also found is that a different strategy has improved water use efficiency in the field. And so you have plants that are growing more, or you've improved growth by improving photosynthesis, but you also improve the plant water status. And so you get more crop per drop, I think, is what we tend to say about that. And we can see, for obvious reasons, why that's becoming more and more important. And so I don't want you to think that the only way to do this is using GM approaches. I think this is another false sort of dichotomy that we've set up in our minds. This is not only possible using GM approaches. Some things will. Borrowing a gene from pumpkin and algae and putting it in a plant is clearly not going to be something we can breed for. But now that we know we can drive 30% yield increases in tobacco, 20% in soybean using a different pathway, then we know that this is something we can start looking for in breeding populations. So if you were here listening to the last talk, you saw some of the wheat populations, and you can see the diversity there. So now we know we can look for this diversity. And this is a really lovely example of rice, where that's already been done. And so looking at photosynthetic diversity in the induction, so time to come up to full photosynthesis from a shade to a sun, right? We don't really think about that limiting crops, but it can limit crop productivity by about 20%. And so looking for variation in that in rice has shown dramatic changes within a breeding population. So now we can start to look for that, work with breeders, and breed our way into, more sustainable crop production. Of course, making better plants is not the only way to fix this situation, and it's not the only thing we're interested in at EPIC. Another major focus for us is manipulating growth environments to maximize productivity. So as we move to a much more urban center, as we move to urban food environments, we know that things like vertical farming or high density planting and aquatic systems are going to play a bigger role. And so understanding what light regimes work best for this, what light regimes can maximize photosynthesis, what types of things we have to consider are a big part of our research. One of the ways that we're doing this is, like I said, looking at light regimes. So plants use the light to power photosynthesis, but not all light is created equal. And so we think light is light, sunlight is sunlight, but actually light has different quality. Plants preferably use red and blue to drive photosynthesis. And if we grow plants under LED lights, we can maximize yield but reduce energy use. And so a major project that my colleague, Professor Tracy Lawson, who's the director of our Essex Plant Innovation Center, is working on, is trying to understand at which point can you maximize photosynthesis? Can you maximize yield and growth of things like leafy salad, lettuces, but also reduce energy use? Because we know for indoor growers, energy uses are going to start to become a major issue, and this year have become a real tipping point for them. And so what we found is that we can vary the light regimes and save about 3 to 5% of energy uses over the course of a day. When you multiply that up to a growing season, it has a significant savings. We also can use photosynthesis to build predictive tools for the industry. So I showed you photosynthesis measured in a chamber. I can manipulate the gases, I can manipulate the temperature. But we can also visualize photosynthesis by using a phenomena called chlorophyll fluorescence. Chlorophyll fluorescence is basically these beautiful photos you see, all right? And the color differences here are the plant absorbing light, using what it can to drive photosynthesis, and then re-emitting a lower wavelength of light back to us. Can our eye see this? No, but detectors can. And so we can use fluorescence-based imaging to understand how plants are responding to different conditions, or whether or not things like herbicide and diseases are impacting plant growth before we can see them ourselves. And so in the research lab, maybe that's not the most important question driving how is photosynthesis responding, but if you have a massive greenhouse and you're growing plants, then you want to be able to have the earliest detection of any sort of herbicide drift or plant disease that you can. We can also use it to detect food ripening, and this is a really beautiful collaboration that, again, is occurring between Tracy Lawson and Vishnu Mohan at the University of Essex to use machine learning to detect fruit ripening and then use robotic picking for soft fruit to come and pick the fruit at its peak ripeness. And so you're reducing waste because you don't have quite as much wasted or spoiled fruit on the vine. And so I think I'm coming to the end of it. I just wanted to give another plug for this integrative approach, because this is part of what really drew me to coming here. You can tell I didn't start my career at Essex, but I'm really happy I've landed here, because it's this integration of biology, technology, and business that's really going to take us to where we need to go. And so with that, please get in touch with us if you're interested in business engagement, or if you are a wannabe scientist, or maybe a scientist that's interested in engaging more with businesses and with policymakers. We're certainly keen to have people on board. And yeah, thank you very much. Thanks to everyone for stopping in. I really, hope you enjoyed it. Thank you.
Thank you, Amanda. That was absolutely brilliant. And the minute you said photosynthesis, I thought to myself, C6H12O6, and I have no idea what that means anymore. Is that glucose?
Yeah, you're not the only
Brilliant.
one. That's glucose, exactly.
Excellent. I bet we have a few questions on GM for Amanda. Who wants to go first? Yes, sir?
Hi, I would be interested, you are showing one plant on the left that was shorter, and then the whole row of plants that was higher. But in my understanding, it was only the stem that had grown. So how do you define quality of the plant?
Yeah, it's a good question, and a major limitation of this model system, definitely, is that this cultivar of plant tends to put on a certain number of leaves, and that's it. And so its stem acts as a storage unit for this cultivar of tobacco. In terms of the quality, that's where the next phase of this research, so it's called the RIPE project, is going next. And so we've moved these out of tobacco, thank goodness, and into soybean and potato at the moment. And so what we find with that is that we can start to look at the nutritional profile of the tuber and see if it's different, which then also as a scientist informs us, what have we changed, right? If suddenly you're accumulating more of something else. But also, have you impacted seed quality for soybean, which is really important, and have you impacted this? And so what we know so far is that we haven't impacted protein to carbohydrate ratios, and that it's harder to work with crop nutritionists than maybe some of us naively thought. Yeah, it's a great question. Yeah, no, it's a great question. Thank you very much.
Ooh, any other questions for Amanda? Well, in that case, maybe I can slightly play the role of the concerned citizen over GM, because it feels like that is definitely an issue here in the UK. When you meet somebody around the dinner table and they have that sort of, bring up something like, aren't you just making frankenfoods in the laboratory? Isn't this going to be a danger for the environment? What is your sales pitch to them as to why that's not the case?
Yeah, it's a good one. I mean, it's a very nuanced question, so there's not an easy answer to this. And there are lots of reasons for people not to trust scientists when it comes to GM crops, right? I think we all know what the gut feeling is here about why it's bad. And I think my answer to this, and the reason that I've come to realize I'm very passionate about pursuing this, is that we are what is it, 2022? So we're about 30 years away from that tipping point of having enough food to feed a mid-century population. It's pretty clear we're going to be in a warmer, higher CO2 world by then. And crop breeding takes about 30 to 40 years. And so we have about one more breeding cycle, barring innovations in speed breeding, which are coming along very well. So, okay, generously, we have two breeding cycles left to really test out solutions to this, to test out solutions to the yield puzzle. Yield is a complicated trait, and it's rarely something that one thing is going to completely solve, even photosynthesis. However, if we don't get that, then we don't want to be in a situation, I don't want to be in a situation where we're looking at 2045 and we have 5 more years where we're really wanting to produce food and we don't have options. And so my goal, my hope, is that no one has to eat these soybeans or these potatoes or this in wheat. That would be phenomenal. That means we've solved the problem. However, I would like to know that there are more solutions there if we're coming to a situation where we need them. I was particularly proud of this work that was showing the temperature thermal tolerance of it, because it means that there's more variation in this thermal adaptation of photosynthesis than we thought there was, and we can start to look to exploit that because we know that these crops are going to start going through increased heat waves and increased, thermal periods. So.
And just finally then, at school and at uni, we all learn in our lanes, and you say an epic, which is a great name, by the way. There's this fantastic collaboration happening. Just tell us how much fun that must be to work with people of all different disciplines.
Yeah, it's really fantastic because it brings you out of your thought bubble. So it's really easy for me to think about photosynthesis or, even one rung out, which feels to you guys like, what is she talking about? But it's a huge step for me to go from thinking about photosynthesis to thinking about yield trials of wheat, or some other crop, right? I wasn't a classically trained crop physiologist or crop breeder. And so that's usually as far as scientists I would feel comfortable going. And then working with someone in math or someone in business helps you to put that different hat on and just skew your perspective into, maybe you didn't see a huge growth benefit, but you saw a massive water savings in the field. And that's really going to matter to people who are growing in more arid climates. Or maybe by changing your red to blue light ratios a little, you haven't seen a huge yield bump or a huge quality bump, but if you saved 3% of energy, that's really going to matter to a business. And it's really nice to come back and have these conversations to think about where can I take this and where can I go.
Thank you. So when you genetically engineer those plants like the tobacco or the tubers, so did you observe any unwanted effects by introduction of the genes, and how did you have to do anything to actually avoid unexpected changes or unwanted changes?
Yeah, it's certainly not as simple as that schematic makes it look. You're constantly looking for failures, and we often find failures. And sometimes that's because your gene has randomly integrated into a gene that's really important, right? So when we put genes into a plant, we don't necessarily control where they end up. That has a very high failure rate. And sometimes, despite the fact that photosynthesis has been studied for 50 years in detail, we still do something that theoretically should have a huge benefit, and the plant suffers from, poor little plant syndrome. So it doesn't do well, it fails to thrive. Clearly, it's not good. And so sometimes we find that too much of a good thing is really detrimental. So sometimes overexpressing certain components of photosynthesis steal energy from different parts of the plant. And sometimes they just do nothing. And I think it's a really important part of doing work like this in a model plant like tobacco. You can go really, really fast. Is to see what's really not going to work, and then you can deprioritize it. And so if you think about it in the same sort of thought that we use for medical testing, right? If you know a drug should work, but then you do it and it doesn't, you want to get rid of it as fast as you can and try to look for something that will. Yeah. Thanks. It's a good question. Thank you.
Thank you. Simon O'Kane, Imperial College London. You talked, the chair talked earlier about the kind of public concerns about genetic modification. What about the concerns that don't surround the safety of genetic modification itself, but the socio-political effects and what, in particular, what do you say to the charge that it gives it gives some companies too much power. So obviously farmers use those companies' services because they are good at what they do, and but how do you create a regulatory framework that protects farmers' interests?
Great question.
It's a great question. It's at the crux of what we're trying to do. So if you want to be involved in shaping a new food system, how do you make a new food system that's equitable? And it's a question I've been muddling with since the early days of my PhD. If you're a plant scientist, you're very much at the coal face of the lab bench, the greenhouse, the field. But I think it's this conversation with people who are, sociologists and psychologists at Essex that want to know, how are people responding to this, and what does it mean, right? And so thinking about this not from necessarily a business perspective. So clearly this work on engineering photosynthesis is funded by the Gates Foundation with support from the FCDO and the American government. And so there are some pretty big implications there for what they're trying to do. And there are some pretty big arguments there with whether or not a company should have that much control over food systems. I think one of the things that motivated me about joining the project was that they're very adamant that what they want to do is set up royalty-free access for farmers in low-income countries. And so many of this sort of climate change aspect of the project was driven because not only are we facing drought and heat stress in the UK, but certainly sub-Saharan African farmers are facing drought and heat stress as well. And so we want to have seeds that are able to withstand those. So it's not a great answer because there isn't a great answer, but I think it's conversation, being open to it, and working with people who are as passionate about policy change as I am about photosynthesis.
Let's embed a philosopher into every team.
Exactly. That's what we need.
Fantastic. Well, we could talk about this all afternoon, but I should probably draw it to a close there because I think the next one's coming up shortly. But Amanda's around for the rest of the day if you want to pick up with her. And yeah, thank you so much for your time. That was absolutely brilliant.
Thank you very much. Thank you all for listening. Really appreciate it.